Our speaker today is Abdullah, and he's been at Simcyp nearly 3 years now, a senior research scientist with a strong background in academic teaching and research in clinical pharmacology and pharmacokinetics. And within the Simcyp team, he's largely involved in large molecules, drug biologics interactions, oligos, parameter estimation, and Simcyp designer. So related to that and his experience, he's going to be talking to you today about his recent tutorial paper, talking about Biologics PBPK platform in the Simcyp designer. So he's got obviously some examples of how it's used and some case studies as well. So without further ado, thank you again, Abdullah, for agreeing to present, and please take the floor.
Thank you, Oliver, and thank you everyone for attending this webinar. As Oliver mentioned, today we're going to talk about the Biologics PBPK platform in Simcyp Designer. And before I get into Simcyp Designer, I generally see modeling tools can be categorized into two categories. The first is the user-friendly tools, and those are usually composed of library of pre-built models that you can use, and they come usually with a graphical user interface that enables you to customize certain model options or parameter values, etc. And when you use this kind of modeling software, like the Simcyp simulator, for example, that reduces the time you spend in coding or debugging your model if you build the model from scratch, which means faster model development. It also makes modeling accessible to users without extensive technical or coding backgrounds, and that lowers the learning curve for modeling development. And it's also easier to review, share, and modify these models across teams, which results in improved transparency and collaboration. However, some users might not fully understand the underlying model mechanics as a result of over-reliance on the user interface. And also having a pre-built model restricts the customization, the level of customization that you can achieve compared to manual coding. And that brings us to the other category of modeling tools, which I call them flexible tools. And these tools are typically allow you to build your own model from scratch, usually through writing a script in some programming language like R, MATLAB, Julia, etc., you name it. And this allows you to implement complex, non-standard, or cutting-edge models. And it also allows you to fine-tune parameters or the logic of the model or even the structure of the model itself to make specific requirements, which enhances the developability and the scalability of these models. However, these modeling tools have much steeper learning curve. They require strong technical skills, often including programming, in addition, of course, to the domain-specific language. And there's more effort typically involved in designing, implementing, testing, and debugging those models from scratch, resulting in longer model development time. And of course, as always with manual coding, there is higher chances of implementation errors or inconsistencies. And that's where Simcyp Designer comes into play. So Simcyp Designer has been designed to bridge the gap between convenience and flexibility. So on one hand, it has a graphical interface with a drag and drop model building environment, which makes model construction very intuitive and user-friendly with no coding required. And at the same time, it allows you to build your own model from scratch. So you have full control over the structure of the model, the parameters, the design of the study, etc. And that results in a balanced platform, which reduces the learning curve of beginners, yet retains the full flexibility for complex models, non-standard models, or cutting-edge models. And what I will show you now is the user interface for Simcyp Designer. So as you can see here, there is a canvas in the middle, which you use to draw your model. And on the left-hand side, you have a palette of modeling objects. So all you need to do is to drag different objects that represent different components or your model into your canvas, and then define their properties and their relationships. For example, you could drag a species, which is typically used to present chemical entities in your model. For example, this could be a drug, an enzyme, a transporter, a target, etc. And there's also nodes that represent reaction, and those could be binding reactions, transport reactions, any reaction that modifies your species. And you can also draw a parameter, which is typically used to represent constant values on your models. For each object that you draw, you have a properties panel, which is on the right-hand side of the Simcyp Designer. In this properties, you can change the name, you can change the initial state, because we are here selected species 2, which is an ODE state. So we can decide what's the value of the initial condition. We can decide if it's a concentration or not, and specify the units. And once you have drawn everything, you can define relationships between those objects. For example, if I want R1 to consume species 1, I can add edge from species 1 to R1. This is called a substrate edge, which means that R1 will consume species 1. And they can add a product edge to species 2, which means R1 will produce species 2. And I can add an edge from the parameter to the reaction, which means that R1 reaction rate expression can use that parameter value. And we also have compartments. So compartments are modeling objects that contain other model objects. So I can move my model into the compartment, which tells Simcyp Designer that those species, especially if they are concentration species, are contained within this compartment. So it can do the appropriate calculation to convert amounts into concentrations and vice versa. So let me show you a more concrete example of a one compartment PK model, which I guess that all of you will be familiar with. So what we have here is a compartment called the central compartment, and this has a volume of 5 and a unit of liters. So a compartment that has 5 liters volume. And inside the compartment, we have the drug, which is a concentration species, has a unit of milligrams per liter, and an initial state of 0. And then we have dosing object, which specifies the dose of this drug. And since we are dosing in the central compartment, this corresponds to IV dose. I'm choosing the bolus options, amount of 0.1 and the unit of grams. And what will you notice here, that the dose unit is in gram, but the drug concentration is in milligram per liter. So the units here are not consistent, but this is not a problem if you are using Simcyp Designer, because it does the unit conversions automatically for you. And then we have a reaction. We have an elimination rate constant and a reaction that eliminates the drug at a rate of K elimination times the concentration of the drug. So if I go to edit grids, that will show me the ODEs of the model. So this is a one-state model. So we have only one ODE that describes the concentration of the drug in the central compartment. So you don't really need a graphical user interface to build a model that is simple like this, because it's one ODE. You could have written it in any language, but you can imagine the unit conversion, the amount of coding required to call an ODE solver to get the solution, to post-process the solution. Imagine that on a model that has 200 or 300 ODEs. It can be really complex to type manually, and I will show you examples of this kind of models very soon. But you can imagine how complex it would be to type it manually, and how simple it could be to do it with a graphical interface. So let me go ahead and run the simulation. So I'll go to simulation, simulate, run. and that will run the model and give me simulation results. And as you can see here, what's what expect from first order elimination. The drug concentration falls linearly on a semi-log scale, and it starts an initial condition of 20 milligram per liter, which if you can do the calculation in your head, it has done the unit conversion and calculated initial concentration correctly for you. You can select the states that you want to output, the parameters that you want to output, and you can go to action and select the output format. You can save the outputs as Excel sheets. You can save them as CSV files if you want to import these outputs into some data processing software and process them further. And you can save it as a database, and those databases can be opened within Simcyp Designer, as we will see in a minute. So I'm going to go ahead and save them as Excel, just to show you how the output looks like. So what it's doing now is it's opening Excel to save the method. Here is the output. Those are the states, and those are the parameters. We have only two parameters, central volume and elimination rate constant, and both parameters are constant in time, so they don't change. And we have one state, which is the drug concentration. This is exactly what you would expect from a one compartment PK model like this. So what if we want to build a bit more complex model? So let's say we want to build a minimal PBPK for biologics. So here is an example. This is a minimal PBPK that describes the PK of endogenous IgG in humans. And as you might have felt, it's overwhelmingly large model. And this is just a minimal PBPK, not even a full PBPK. I'm not going to go into details of this model and explain it, but what I want you to get is, you probably have already got it, it's not going to be easy to build biologics PBPK model. So the question now that you might be asking, why isn't it too complex for biologics PBPK model? So in generally, biologics are large molecules. They are hydrophobic. They have large molecular weight, so they cannot passively permeate through cell membranes. And that's why when we describe their PBPK, we always use a permeability limited organ structure. So all organs in a PBPK model, whether it's a minimal with one tissue or a full PBPK, will have a four compartmental structure like the one that you can see, with vascular, endothelial, interstitial, and intracellular, with various processes describing the transport of the drug from the vascular to interstitial and vice versa, binding to FcRn for monoclonal antibodies, uptake into the cell, blood flow, venous outflow, lymph flow, etc. And to build the full PBPK model, we combine those four compartmental structure. We replicate this to represent different tissues and combine them into the full PBPK model. So each rectangle here represents an organ, which is made of the same four compartment structure that we have seen on the previous slide. So if you want to build a standard monoclonal antibody full PBPK model, when I mean standard, I mean there is no TMDD, there is no IgG competition, there is no any fancy stuff, just a plain map model. You will end up with 236 ODEs and more than 1,300 parameters and more than 1,000 algebraic relationships, in addition to 300 plus reactions. So you can imagine how complex building that model can be from scratch, or building it manually through coding. And if you want to make that model a cross-species model, so you want to have models for preclinical as well as clinical, which is not uncommon in biologics drug development, then you have to multiply that effort by the number of species. So if you want 4 species, for example, then you will end up with around 1,000 ODEs, which is doesn't seem to be feasible to be done manually. And this is what we have addressed in the Biologics PBPK platform in Designer. So what we thought about is that we want to automate Biologics PBPK model building, and this automation exploits the redundancies that we have in the model. So the first thing that we can use to our advantage to automate this model building is the structure similarity across tissue organs. So almost nearly all organs have the same 4 compartmental structure, which means that we don't have to build those 4 compartments for every single tissue. So we can use the so-called module, where you define the structure for one organ and then reuse this module to represent different organs, which I will show you how we do it in Simcyp Designer. The other thing that we can use to our advantage is that the whole PBPK structure, which I have shown in this slide, is exactly the same in the 4 mammalian species or 5 or 6 mammalian species that are commonly used for preclinical drug development. So all animals have, or mammalian species have lungs, hearts, livers, guts, etc., and they are connected in the same way. So the structure of the whole PBPK model doesn't differ from one species to the other. And to use that advantage, we have separated the model structure from the model parameters, which means when you build the model, you define the structure, how the compartments, how the species react, what are the parameters, how these parameters are calculated, and then how they are used. But we don't specify parameter values. We don't have to until it comes to the step where we simulate the model. Then we can parameterize the same structure for different species. So if we use the physiological values for humans, the same model becomes a clinical model. If we use the physiological values of, for example, mice, rats, monkeys, then the model becomes a preclinical model. So we need to build the structure only once, and we can use it multiple times for different species. And I will show you how we do that in Simcyp Designer. So this is what we call the platform model. So the platform model is what I have just called the standard monoclonal antibody model. So we have here the generic tissue module. So this module defines the generic tissue structure. And then we have the so-called instances. So instances are basically copies of the generic tissue module, but they are not just Control-C, Control-V copies, also known as static copies, but they are dynamic copies, which means they are actively listening to the generic tissue structure. So if I change anything in the generic tissue structure, it will be automatically updated in the full PBPK model. So it's a dynamic rather than just a static copy. And inside the generic tissue structure, I have 4 compartments: vascular, interstitial, endothelial, intracellular, just as you have seen in the diagram. And below that, I have the parameters module, where I define how my parameters are calculated, what are the relationships between those parameters. And as you can see, I can easily block or open up any modules to make visualization easier. So this, what you see now, is just the structure. There is no any parameter values defined yet. So in order to simulate this model, I need to define what species I want to use it for. So I can go to simulate run VPOP this time instead of run. And that will give me the VPOP manager. So VPOP is just a CSV file that contains alternative parameterizations of the model. So in this file, for example, I have 4 species. So each row has the parameter values for one species. So I have here a row for mice, rats, monkeys, and humans. I can select whatever species I want. I can select more than one species. For example, I can select here rat and human, click sample selection, then run sample. Or if you are interested in all species, you can click run all. I'm not going to run it now to save your time. I have run it before this seminar and saved the output database. So I can open QSP results database, and that gives me the results of the simulation that I have done just before this seminar. I can look at different species, different drug species I see. For example, here you can see the drug concentration in the vascular compartment of the adipose tissue, and you can see the simulation results for the 4 species: mice, rats, monkeys, and humans. So now you might be wondering, how on earth could you call this user-friendly? It seems very complicated model, but in fact, it is not. And I will show you now how I turn this, let's call it template model. It is template because it's easily editable and easily convertible to different kinds of models. So how I convert this template model into an oligonucleotides model. So let's grab the oligos file. So what you can see here is the exact same model that was on the previous screen, but what I have done is just I deleted all reaction related to FcRn binding because oligonucleotides don't bind FcRn as opposed to monoclonal antibodies. And I have added 2 assignments to calculate how much of the drug is bound to plasma protein and how much is free. And this calculation doesn't exist in the monoclonal antibody model because MAPs don't generally bind plasma proteins, whereas oligos PK are highly affected by protein binding. And that's why we have added this component. And the final modification is that we have added binding to ASGPR receptor in the interstitial compartment because some oligos are conjugated to ASGPR ligands. So we have added receptor-mediated endocytosis reactions. So we haven't touched, in this model, we haven't touched the full PBPK model. So the full PBPK model is the same. But actually what is happening behind the scene is that each of these tissues reflects the new generic organ structure. So the whole PBPK model, instead of being a MAP model, it is now an oligonucleotide model. And you can see in this slide how this model performs. Those are the model predictions for various tissue concentration in rats and in mice versus observations, which are data we have collected from literature. And this model has been previously published in JPKBD, and the publication is shown on the screen. If you are interested in more details, please refer to this publication. Another case study to show you how flexible and how easily flexible this model can be is developing a model for IgG in pregnancy. So this is the IgG pregnancy model. So the genetic tissue structure is exactly the same as the MAP model because IgG behave exactly the same as MAPs. However, what we have changed here is the full PBPK structure. Since we want to model pregnancy, we want to add instances or copies of the generic organ structure that represents the placenta, the fetus, the fetal lymph node, and the fetal blood flow, arterial and venous compartments. And we connect those tissue instances. We connect those tissue instances to the full PBPK model to represent the blood and lymph flow within the fetus and within the mother's PBPK model. And then when we, of course, we would have to provide new parameters for these models. For example, the placental blood flow, the placental volumes, the fetal volumes, and fetal blood and lymph flows. And once you parameterize this model, we can compare the IgG concentration prediction in both the mother and the fetus. And as you can see in this slide, the model very well predicts the maternal and fetal concentration. So the idea here I want to give you is not about how good the model predictions are, but how easy it is to turn our platform model or template model, which is MAP model, into any other large molecule modality. And we have recently published a tutorial, a full tutorial on using the Biologics PBPK platform in Simcyp Designer, and the publication is shown here on the screen that you can refer to if you are interested in more detail. If you are even more interested, there is a workshop that we are running. It's an online live workshop in November, in which we will go over all details of building and using the Biologics PBPK platform in Simcyp Designer. If you're interested in registering in this workshop, you can do so by going to simcyp.learnupon.com and then register in that workshop. And with that, I would like to thank you all again for your attention and open the door for any questions or comments that you might have.
Thanks, Abdullah. That was really clear. And yeah, we can take any questions if anyone has them right now. Just while we're waiting, so can these models be adapted or scaled up for QSP applications?
Yes, actually, that is one of the main advantages of using QSP Designer is that you can easily link it to QSP model using different ways. The first way you could do is that you can just build your QSP models in the same workspace as your PBPK model, and then link the driving concentration, which could be the drug concentration in any tissue compartment that you're interested in. Or the other way is through the imports feature. So the import feature allows you to import another model into your workspace. So what you could do is that you can build the QSP model in another file and then import it into this PBPK model and then link them. Or you can do the other way around. You can import this PBPK model into your QSP model and use the drug concentration in any tissue compartment as the driving concentration for your QSP model. So it's perfectly suited for PBPK QSP modeling.
Oh, that's great. Is there anyone else with a question? Can't see any other questions.
If you have any question, feel free to raise your hand or unmute yourself or type the question in the chat.
If not, yeah, you can always reach out to Abdullah as well after the session as well. Okay. I can't see any other questions coming through. Thanks again, Abdullah. Just to flag that the next webinar is on mechanistic modeling of drug release from PLGA-based implants, and this can be focused on the case study of dexamethasone solid implants. And that will be on Tuesday, the 26th of August. And Jean probably be back in the driving seat to host that. So thank you, everyone, and take care.